So to prevent from the cytotoxic T cell, some of the virus will present, virus will be decode from the false MSC class 1 that will be actually recognized by this NK cell and can be killed. So that will also be understood. If this NK cell cannot kill any cell that has the normal expression of the MSC class 1, or say they have the normal HLA-E. So if the HLA-E is there, so means it is a cells has normal MSC class 1, that will not be killed by the NK cell. So if there is abnormal, if the MSC class is down regulated, if there is abnormal MSC class 1, then what will happen? This will actually, NK cell will goes and kill there. The killing mechanism will be the same. The perforin and granzyme, once they get attached, they will release this perforin that will create the pore inside the cell. Then they release the granzyme will go activate the caspases and kill that cell. So this is the one of the mechanism of killing your about the CD8 cell, NK cell. Now talking about the ADCC. ADCC is the final mechanism of cytotoxicity which bridges humoral and cell-mediated effectors system in the body, is an antibody dependent cell mediated cytotoxic. This is called ADCC. So this is the combination of, say, ADCC is not a cell mediated immunity or humoral immunity. This is the combination of both the cell mediated and humoral immunity, because they are using the interferon gamma cause class switching of the immunoglobulin G, and this class immunoglobulin G has the property of binding to the damaged cell as in the fragment of antigen binding site, and their tail portion will bind to the phagocytic cell, and in this way they can digest or kill the cell. This is called antibody dependent cell mediated cytotoxic. A number of cells with cytotoxic potential, that NK cell, macrophage, neutrophil, eosinophil, have a membrane receptor for this Fc region of the immunoglobulin G, that is the CD16, where immunoglobulin G is especially bound to a target cell. The cytotoxic cell can be bind to the free tail and subsequently cause the lysis of the cell. Although these effectors are not specific for antigen, the specificity of the IO types of the antibody detects their cytotoxic. This mechanism of target killing in case may involve the following, that is glytic enzyme, tumor necrosis factor, and also the perforin and granzyme. So what will happen? You can see there is the neutrophil, macrophage, this is the NK cell, eosinophil. They are attaching to the tail region of this immunoglobulin G, and then antigen binding part is attaching to the target cell, that is damaged dying cell, and then this will release this lytic enzyme, perforin, granzyme, and TNF that will damage and cause the apoptosis of the cell or activate the caspases and cause the death of your cell. This is called antibody dependent cellular cytotoxicity. So one more important thing is, when you talk about the cell mediated immunity, it is actually going to kill the whole cell, our own cell that has been infected with the intracellular pathogen. So the antibody cannot enter inside our cell, so we don't have any option and we need to kill that cell, but that cell is actually dying, disease. So it is wise to kill that cell because they contains the intracellular, which we can say tickling bomb inside it, so intracellular pathogen inside it. So this need to be cleared. But you have to understood that this cell mediated immunity doesn't kill your own cell, because this normal cell cannot be killed by NK cell or cytotoxic T cell because they have normal MSC class 1 protein. Okay, so hope you have understood, and you have also understood why transplant are actually rejected, because those transplant contain this molecule, the MSC class 1, that is from the other portion that is obviously become foreign to me, and if my CD8 cell will recognize any MSC class 1 as the foreign, then it will obviously going to kill that cell and cause the rejection of the transplant. So hope you have understood the cell mediated immunity. It is not so much, you do not need to go deep inside it. It is just killing the cell, the disease and dying cell by your own immune system. Thank you.